welcome back to part 3 of acid, bases and salts. So today in this video we are going to talk about the indicators of the chapter, that what all are the indicators that identify that whether it is an acidic substance or a basic substance. So let's begin with this very interesting indicators video. So now let's talk about indicators. Now let us know firstly that what are indicators. Indicators is something that which indicates us. For example, in our daily lives, we use our cars. Okay, so we have indicators in our cars. Whenever we have to take a turn from a middle road, we show the indicators to the other persons who are driving, because no one should come in the middle as we are taking a turn, or it should have an accident. That is why we have indicators in the cars as well. So that is why we use indicator to indicate someone or to alert someone that this is going to be happen in next few minutes or next few days. So indicators is something that indicates us, or it indicates us with that which substance is it, it indicates the difference between each other. So now let's talk about all the indicators in it. Now our first indicator is litmus paper. As I told you that litmus paper is used to identify whether it is an acidic substance or a basic substance. So there are two types of litmus paper, red and blue. So we use blue litmus paper whenever we put blue litmus paper into an acid. It changes into red litmus paper and it shows that this is an acidic substance. But if we talk about bases, we take red litmus paper and put it into the bases, and it converts into blue litmus paper and it shows that this is a basic substance. So let me revise that what are the indicators in the litmus paper. Litmus paper are two types of blue and red. Blue is used to put it into the acidic, so it converts into red litmus paper, and red is put it into the basic, so it converts to blue litmus paper. So these are the points, these are the first point of litmus, that how do we indicators. The first indicator is our litmus. Now let's talk about our second indicator. Second indicator is pH scale. We talk about pH scale. pH scale is about 0 to 14, and where 7 is the neutral, from 0 to 6 is the acidic lane, and from 8 to 14 is the alkaline lane or the basic lane. So let's talk about how do we use it. How do we use this pH scale in our daily lives? So if we need something to identify, we are usually in lab. So pH scale is usually used in labs. So if we talk about pH scale in daily lives, there is a strip. It is in yellow, light yellow. So we put we have to take there is a water. We put that pH paper in that water, and if after 2-3 minutes we have to just take out from the water and check the color, which color it is matching with. If no one, so then it is a neutral. If it is matching on the number of 7, if it matching colors which is on 7, it is a neutral substance. Yes. So water is a neutral substance. So if ever we put a pH paper in a water, it always show the neutral. Now let's talk about acidic substance like HCl. Whenever we put a pH paper in HCl, it will show in the lane of acids. It will always show in the lane of acid and nearest to 0, because HCl is a strong acid. Now if we talk about bases, whenever we put a pH paper in potassium chloride, sodium chloride, so it will always show in the lane of alkaline or the basic lane, and most of the strong basics will be in nearest to the 14 number. That is why these are the strong bases. Now this was the second indicator. Our second indicator is pH scale. Now let's talk about the third indicator, which is turmeric paper, turmeric solution. Okay, now how do we use this turmeric solution? How do we make a turmeric paper to take out the solution, whatever we have to take out it? So what basically we have to do, we have to take a filter paper, a big filter paper, means filter paper when we filter something. So that filter paper, and we have to make the paste of a turmeric mixing turmeric and a little of water. After making the paste, we have to spread it on that filter paper and keep in the sun, keep on the hot on warm place for 2-3 hours, and after that it will be dried, and then you can use it and you can take it out from that filter paper and you can use it in your life, in your daily routine, that how to use the turmeric paper in the daily life. What you will do that you can paint something on that paper, like you can use vinegar. You have a brush, put that brush into the vinegar and write something with that brush on the turmeric paper. It will just change into brown or a little reddish brown color, because it shows that this is a basic substance. Whenever we put the turmeric paper used in basics, it get changes into red, and acid, it get changes into red, and whenever we put into basic, it remains the same. Now let's talk about the third. So our third indicator was turmeric paper. Let's talk about the fourth indicator. Fourth indicator is the flower. China roses, China flowers. So how do we use that? Let us talk about how do we use China roses, China indicators, China solutions. So first we take any China rose and take 2-3 petals of that and put into the water and take out, put into the water for 2-3 hours or 3-4 hours, and after that the petals will remove its color into the water and the water will become a little baby pink, and take out all the petals, and after that put that solution, that China rose solution, in any acidic. It will turn into magenta, dark pink. It will turn into, whenever the China rose solution reacts with acids, it turns into magenta. And whenever the China rose react with bases, it turns into light green. It turns into green, normal green. It turns into normal green. So when the China rose reacts with acids, it turns into magenta, and when it reacts with bases, it turns into green. This was the fourth indicator. Our fourth indicator was China solutions, or the China rose solutions. Let's talk about the fifth indicator, which is phylloxetine. Phylloxetine is an indicator in which it is a indicator in what we do. It has a transparent, its solution is a transparent, it does not contain any color. When we put that phylloxetine in acids, it gets changed into baby pink, and when we put phylloxetine into bases, it remains the same. It does not have any change in the bases. Now a fifth indicator was phylloxetine. Let's talk about our sixth indicator. Our sixth indicator is methyl orange, and from the name we can understand that the indicator solution is orange. Yes, the indicator solution is orange. Whenever we put that orange, methyl orange solution in an acid, it gets changed into dark orange or something, a pink, dark red, or mix of orange and red, and whenever it gets mixed with bases, it remains the same. It remains the orange only. So our sixth indicator is the methyl orange. Now let's revise what are the indicators, what are the 6 indicators of this chapter. First is litmus paper, then second is pH scale, third is turmeric paper, fourth is China rose solution, fifth is phylloxetine, and sixth is the methyl orange. So these 6 are the indicators of acid bases. How do we identify these acids and bases? So if you like this video, please subscribe, share and like. Bye bye.